research scientist at Marshfield Clinic. I'm here on behalf of Marshfield Clinic, but also the All of Us Research program. I don't know if you're familiar with All of Us, but my goal today is to talk a little bit more about the science that's coming out of the program, not necessarily here to enroll people. Obviously, if you're interested in enrolling, and if you haven't already and want to do so, you still can. But again, my goal here is to really show the wonderful things that this program is already doing with the people who have already enrolled. Now, what I will be talking more about is cancer, which I think we're all very sensitive to. I'm sure that every one of us has known someone in our life, close or otherwise, who have been touched by the disease. And I'm also going to be talking about telomeres. And all this work is done in part through the All of Us Research Program, but it was also done in a biobank at Marshfield Clinic called the Personalized Research Project, I'll call PMRP, which was a precursor to the All of Us Research Program, again, for what I'm here for. And so a little bit more about me. I grew up in Appleton, Wisconsin, went to school at University of Wisconsin-Eau Claire, got a job at Mayo Clinic, was fortunate to be able to get into their graduate programs a little afterwards, and came here. And most of my research focuses on genetics and understanding human disease and advancing concepts of precision medicine. And what I mean by precision medicine is that is a strategy in healthcare where we're moving away from that one-size-fits-all strategy to care to one where we begin to treat people more as unique individuals. That's where the precision part comes. Because as I think we can all realize, every one of us is different, both genetically, our health histories, our family histories, our occupations, our education, our religions, all these factors who make us unique in who we are, also impacts our health. And that's the research I'm passionate about, and that's also the foundation of the All of Us Research Program. So I'm going to be talking about telomeres, and I'll stand up to describe this. So there's going to be a number of technical terms that I'll be describing today, and I recognize some of it's going to be alphabet soup. So hopefully I'll be able to describe it in enough terms so that you can understand what I'm trying to communicate, but at any point you have a question, don't hesitate to reach out. So the first term is telomere. Telomeres are protective DNA caps that we have on our chromosomes. I'm showing you here a picture of a chromosome, and at the tips are those telomeres. What's unique about telomeres is they are the longest when we're born, and they are the shortest when we die. So you have a sperm, an egg, they combine, you have the longest possible telomeres, and then that cell starts out with one cell, then goes to two cells, it splits. And at that point, the telomeres get incrementally shorter. 2 goes to 4, gets incrementally shorter. And over time, those telomeres get shorter with every time a cell divides. So imagine me as a chromosome, and my telomeres are my arms and legs, and all the important stuff is in between. So I start off with the longest, and then one cell division, okay, I lost my fingers, and then lost my hands, and maybe by the time I'm 50 years old, I'm just a stub. I still have some telomeres, but still everything else is protected. Once the telomeres get incredibly short and starts degrading into the body of the chromosome where all the important information is kept, all of your genes, that would be bad for the chromosome, that would be bad for the cell. And when that happens, there's biological mechanisms that cause the cell to stop dividing and eventually die. So that's what the telomere is, and they're really important to protect all that information in the chromosome where all our genetic information are. So what's really cool about telomeres is they've been called a biological clock. So you can measure someone's telomere and you can guess approximately how old they are. But not every 50-year-old has the same telomere length. And there's a lot of factors that influence that, and I'll talk about that in a second. So what I'm showing here, I'll just stand. So we have a chromosome with their telomeres, and this is called the distribution curve, and it's graphing out, let's say, telomere length in relationship to the proportion of a population. Most people have average telomere length. There are a few people that have exceedingly short telomere length. And then there are a few people that have exceedingly long telomere length. But again, most people are somewhere in between. The people that have exceedingly short telomere length have a whole wide variety of diseases that they can develop. That includes bone marrow failure, kidney fibrosis, liver fibrosis, pulmonary fibrosis. Essentially, these people are prematurely aging because they have abnormally short telomere length. Those are often due to genetics. So there are genes that maintain our telomeres. They're buried within, but if there's a mutation, a genetic variation in one of those genes that you've inherited, these people can develop these, usually these are called telomeropathies. And they can be fatal, and they typically are. So in your 20s and 30s, those people unfortunately will die from their disease because they have exceedingly short telomere length. And you might imagine, again, if your cells just have really short telomeres, they're just dying because they just got too short. Now, over the last few decades, people have thought, this is good. As I mentioned before, telomeres kind of predict your age, and there's a lot of factors that impact your telomere length, and so if you have really long telomeres, it's thought, well, you're living a healthy life. So a lot of factors, including your diet, bad diet results in short telomeres, lack of exercise results in short telomeres, exposure to a lot of bad chemicals results in short telomeres. So it's been thought for well over a few decades now that people who have inherently long telomeres are probably very healthy people and might live a long life. But what we're going to show is that may not be the case. There are certain factors that might be bad when it comes to long telomeres. Okay, so I mentioned a little bit ago that there are genetic factors that can predispose people to have really short telomeres. And a lot of that was done in family studies. So you identify a family, and a number of people within that family share this disease profile. They have lung fibrosis, kidney fibrosis, or bone marrow failure, and a variety of other things. You recruit them into a study, and then you look at their genome, and then you hopefully can map the gene that's causing this family's disease. And they've done that with a few families to identify a few genes that are associated with these really short telomeres. Those studies are really hard to do because it's not enough to recruit just one person. You have to recruit a whole family to do this type of research. And so you can imagine if you have a family of 5 where half of them say, no, I'm not interested, then you may not be able to do much with that family. And these families are exceedingly rare. So people have done now, they've moved away from these family-based studies to population-based studies. And what I mean by that, these are people who are all unrelated. And they're called genome-wide association studies, GWAS. It's going to be the first alphabet soup that I'm going to talk about here. And I'll describe a little bit how this works. So we're in a room right now with 4 people, 2 men, 2 women. And let's imagine a GWAS study that we want to do within this room. Let's say we have a disease group. That's going to be the men, because we all know the men are the inferior race here. And then we have the controls, which would be the women. What we would do is we'd look at genetic variant 1 on chromosome 1 and determine whether or not that genetic variant is more common in the women versus men, in your disease versus undiseased, your control group. And you calculate a level of significance using statistics, and then you move on to variant 2, and you do the exact same thing. Is that different variant on chromosome 1 that happens to be just like right next to the first one, and you ask, well, is that variant more common in your case group versus your control group? And then you do the same thing for variant 2, and then before you know it, you march across all of chromosome 1, chromosome 2, 3, 4, 5, 6, all the way to 23, which encompasses 10 million or more genetic variants. And you have this statistical test for each one of these variants. Now, people have done this type of work for studying thousands of diseases now. We've done this work for studying telomeres. So using Marshall Clinic's Biobank, which I talked about a little bit ago, it's called PMRP, the Personalized Medicine Research Project. We enrolled 20,000 people. This was back in early 2000. These were people who generously said, yes, you can have my DNA, and yes, you can use my health information. And that allows scientists to study any disease that they can pull out of the health records and combine it with genetics. So for this population, I had all their DNA, and I measured everyone's telomere length. I adjusted out the age component, and then just say, okay, what the variation in telomere length that we're observing is explained by variant 1, variant 2, variant 3, up to 10 million variants. And this was our result. This is called a Manhattan plot because people associated with the skyline of Manhattan. What I'm showing you here are 10 million data points across all of chromosomes. And then across the vertical line is a statistical measure of significance. The higher that number is, the higher the dots, the points on this graph, the more excited we are as scientists. In reality, anything above this red line considers statistical significance. And what we found is a dozen or more genetic variant, we found hundreds of genetic variants because each one of these dots represents an individual genetic variant, but all those variants map to well over a dozen different genes that are associated with just normal variation of telomere length. I showed you that distribution. Go back a couple. I showed you this distribution, and this is what we did. I mean, we measured 20,000 people, and we just graphed out where they fit, and we have a measurement, and then we associate that, link that to each one of the 10 million variants. And this is what we got. That was done in Marshall Clinic patients that was initiated, these people enrolled nearly 20 years ago now. So I've shown now we have a dozen or more genes that are just associated with normal variation of telomere length. I'm not saying these are people who have exceedingly short telomere length or exceedingly long telomere length. These are just normal variation. We following so far? Mm-hmm. Okay. The next question we had is, well, are there any diseases that are connected to telomere length? I mentioned before that short telomere length has been characterized with poor health, such as lack of exercise, poor diet, exposures to certain chemicals. There have been literally thousands of papers and studies looking at telomere length, linking it to human disease and other factors. There's even a paper that published that the closer you live to a busy highway, you're going to have shorter telomere length. To the discussion we had before, maybe, and was it St. Louis or Louisville?
Louisville.
Yeah, maybe people that lived where they planted all those trees might have longer telomere length than people that weren't in that, as an example. Mm-hmm. People have done literally thousands of these studies, and they typically focus on one disease and make these associations. The beauty of working with a biobank, like the Personalized Medicine Research Project, our 20,000 patients, we can study all diseases simultaneously. And that's doing what's called a phenome-wide association, PheWAS. So I talked about GWAS. GWAS, we're mapping genes to disease. PheWAS, we start with the gene, and now we're looking to see which diseases are connected to the gene. So imagine a needle in a haystack. We have two haystacks. One haystack of genes and one haystack of diseases. And we're trying to find that proverbial needle in either of them. In the haystack of genes, that's the GWAS. In the haystack of diseases, that's the PheWAS. So we measure something, a gene, or in this case, telomere length, and we ask, when we have electronic health record, we know who's diagnosed with breast cancer, colon cancer, rheumatoid arthritis, osteoarthritis, multiple sclerosis, ingrown toenails, plantar warts, COVID-19. We know all that, and for every one of those diseases, we can link that to the gene or the genetics, in this case, telomere length. And we can do that for one disease, we can do that for two, and we just march across all the diseases. Before you know it, you have a test where you studied 1,000 diseases simultaneously and linking it to your genetics, telomere length. And again, we've done that before, and we did this in PMRP. This is, again, our Manhattan plot. No longer are we talking about millions of genetic variants. In this case, we're talking about 1,000 or more diseases across the horizontal. And again, this is still level of significance. Anything above this line is statistically significant, where scientists get excited about it. There is a lot going on here, and we did it in a single experiment. We did it with 20,000 people. What's interesting about this is nearly every single one of these data points are associated with short telomeres. So increased risk for just about every one of these data points is associated with short telomeres are associated with increased risk for some infections, mental disorders, respiratory disorders, digestive disorders, all these things. And that's consistent with what's been published in the literature for a few decades now. When they show all these studies, it's always short telomere length, bad, associated with disease. And I said that in the beginning, but very little has been studying long telomeres. Is there a negative consequence to long telomeres? And what's interesting, there was one exception to the rule that I talked about, where everything was associated with short telomeres. These were the diseases that were only associated with long. Everything else was short, and they're all related to tumors. Still following? Okay. So we had a hypothesis that we wanted to test with all this data. And that hypothesis was that people with long telomeres, specifically people who have a genetic predisposition to long telomeres, these aren't people who don't smoke and exercise well and eat well, who might have longer telomeres. These are people who have a genetic predisposition to long telomeres, that they may have a tumor predisposing syndrome. There has been a lot of study in cancer research. We know a number of genes that are associated with these cancer syndromes, these tumor syndromes, whether it's the breast cancer genes, there's colon cancer genes, there's endocrine tumor genes, but there have not been many new discoveries in this realm when trying to understand people that have multiple different types of tumors and variable types of tumors for that matter. So in order to test our hypothesis, we then leveraged the All of Us research program. Everything I talked about before was what we did here at Marshfield Clinic with our own patients. Now we're going to move to a new population. Marshfield Clinic population, our biobank was 20,000. The All of Us research program
is trying to enroll 1 million or more people. So you can imagine the scope at which you could do these experiments when you have so many more people, because there's only so much variability. We have 20,000 people who are all unique. Imagine what you can do with 1 million or more people who are all unique, including at the genetic level. And so what we did, step by step, is we took all the genetic data that we know that are important for telomere variability, and we condensed it into a polygenic risk score, a PRS. And I forget, please forgive me for alphabet soup, but I'll try to walk you through what each one of these terms mean. But now we have this PRS, we condensed it, and then we did a PheWAS on it to identify here the diseases that are associated with telomere genetics. And then we wanted to condense all the phenotypes into a risk score. It's called a phenotype risk score. So you can imagine that as like a fingerprint characteristic of a disease. Imagine, as an example, I think many of us are familiar with cystic fibrosis. Cystic fibrosis can be a devastating condition. People with it includes male sterility, hypoxia, and your fingernails can get rounded and just unique fingernail patterns. You're going to have numerous types of lung infections, chronic bronchitis, a variety of other types of conditions. Now, each one of those by themselves doesn't really characterize cystic fibrosis, but when you look at them all together as a total syndrome, then you understand who does and doesn't have cystic fibrosis. That's the same thing with the phenotype risk score. We're taking all the diseases that are associated with telomere length and turning it into like a fingerprint for that disease. That's what the phenotype risk score is. And then our goal was to identify new genes that are, and rare genes that are high risk for people who have these tumor syndromes caused by long telomere length in this case. And then we actually validated the biology showing that indeed they're associated with long telomeres. So I'll walk you through each one of these steps. So first thing we did is we wanted to generate our genetic risk score, our polygenic risk score, PRS, for long telomeres. So I showed you this Manhattan plot. What we did is we took all the important genes and all the important variants and we condensed them into a single score. So imagine we have a room here of 4 people. Maybe one of us had, so there's like a dozen, one of us might have a genetic variant in all 12 of these. Unlikely, but maybe. That would be, this is the distribution of scores. Someone might have all the variants. There might be someone in this room that has none, but chances are we're somewhere in between. Someone might have 10, someone might have 5, someone might have 3. Most of them will, again, be somewhere in the middle. That's our polygenic risk score that we did for everyone. And we did that in over 500,000 people that were enrolled in the All of Us research program. And what we did with that polygenic risk score for telomere length, again, this is just normal variation of telomere length and the genetics of normal variation of telomere length, we did a PheWAS off of that. Trying to identify what diseases are associated with telomere length genetics. And this is our PheWAS result. Another Manhattan plot. It might look slightly different than the last time, but again, here's all the diseases, well over 1,000 different diseases. Here's our level of significance, and anything above this dotted line we're excited about. Every single one of these data points are all linked to predisposition to long telomere length. All of them are connected to a tumor type trait, both benign and malignant. So that was very consistent with what I showed before. So before we were actually measuring telomere length itself. This, we're looking at the genetics of telomere length. You understand the difference? And when we look at the genetics of telomere length, everything is associated with long telomere length, and they're all related to tumors. And we did that in the All of Us research program with hundreds of thousands of people who were graciously willing to participate in the study. Now what we want to do is condense all that Manhattan plot data for the phenotype risk score into our, we want to condense this into our phenotype risk score. So all of these data points we condense into a score, and again, similar to that cystic fibrosis analogy, if you have a group of people in a room and they have variability of traits consistent with cystic fibrosis, who's going to be the person who most likely has cystic fibrosis? Is it the person who just has chronic bronchitis? Or is it the person who's going to have chronic bronchitis and male sterility? Or is it going to be the person who has chronic bronchitis, male sterility, a history of infections, hypoxia? It's going to be the person that has all the different traits. And that's what we did here, again. And this is the distribution of that type of information. Most people have none to one or two diseases consistent with here, but there are people who have all of them. Those are the people we're most interested in. Those are the people likely to have a genetic predisposition to this tumor syndrome. That was what we were testing. So in order to test that, we looked at the genetics of telomere length, the normal variation of telomere length, and then we looked at the phenotype risk score. And what this is just graphing out is the score, the polygenic risk score, the genetics of telomere length versus the phenotypes associated with telomere length. And you can see here that people who have a low score, low genetic score, typically have a low disease score. People who have a high genetic score typically have a high disease score. That's what you would expect. That's based off just normal variation. So if you inherit multiple variants associated with long telomere length, you might have this tumor syndrome. The next thing is we wanted to look for novel variants. Again, these are the rare variants. We take our distribution here and we're just looking at all the genes and all the variants, focusing on the rare variants, and this is another Manhattan plot. Looks a little bit different than the last ones, but in this case, again, we have millions of genetic variants across the human genome, and then we have level of significance, and anything above this line, we say, wow, this is cool. And what we found is 13 different genetic variants across 13 independent genes. The vast majority of these genes had never been known before to be linked to cancer. So these are new discoveries in the field of cancer that may be related to inheritable forms of a tumor syndrome. There's only one exception to this, and that's ATM. That was the one gene that had been previously implicated in tumor genetics. The vast majority of the other ones were not. So that was also done in the All of Us research program. And it could only have been done in a population like that, where you have hundreds of thousands of people who have provided their genetics as well as the disease information. And it's really cool because they, just like PMRP, which was literally a foundation for, or a blueprint for the All of Us research program. People, just like PMRP, provide information about their health records and their genetics, but then All of Us expands far greater than that, because human disease isn't just about genetics, it's about the environment, and the All of Us program is going well beyond. So I've thus far described our genetics of telomere length, condensed that, we did a PheWAS of that to identify here the diseases or the phenotypes that are associated with telomere length genetics, and identify these genes across the human genome, the typically rare variants that are associated with people who have this long telomeropathy type syndrome. And then we wanted to actually test that biology. What I mean by that is we have the gene, we know the gene, it's connected to this telomere syndrome, or a telomere-like syndrome, because it all started with telomere genetics. We wanted to actually show that these people who have these variants actually have long telomeres. Went to the All of Us research program, So this is kind of cool history. The most expensive and most ambitious biomedical research study that was ever conducted before what I'll be describing before was called the Human Genome Project, where they spent billions of dollars to sequence a single genome. That, as a geneticist, that was extremely invaluable for the research I do today. All of Us research program has a goal of sequencing 1 million or more whole genomes. So we now have all this genomic data. I went to the All of Us research program and actually started measuring the telomere length in people who have the variant and people who don't have the variant. At the very top, what I'm showing you here is just a description, a representation of the distribution of telomere length in people who don't have a variant. It's called wild type. Let's see if I can get a marker. Okay, so this line represents the mean, and then we have, this is where most of the people have their telomere length, and then you have some confidence intervals and outliers. That's what the data points represent. That's just normal variation of telomere length, and that's based off of people who don't have the variant. And then everyone else, these are all the 13 other genes. And each one of these stars represents, those are statistically significant. And what they show is that for 9 of the 13 genes, they're all associated with long telomeres. So I hope that makes sense, because we started with, I showed in the very beginning that people who have long telomeres, where we actually measured the telomere length, are associated with a whole lot of diseases. Most of them are associated with short telomere length, but the ones with long are associated with tumors. We then go to the genetics, so I guess I'll go step by step. So now we map out, here are the genes that are associated, linked to just normal variation of telomere length, and we do a genome-wide association study, a GWAS. We condense that information into a polygenic risk score. Once we have the polygenic risk score, we do a phenotype, phenome-wide association, a PheWAS. We talked about the two needles in the haystacks and the differences between GWAS and PheWAS. Once we do the PheWAS, we now have all the phenotypes associated with genetics of telomere length. We condense that into a phenotype risk score. And then we did a GWAS of that to identify these new high-risk genetic variants associated with the genetics of long telomere length, and proved that they were actually associated with long telomeres. So, what are the conclusions of all this work? Started in PMRP, we've now migrated to the All of Us research program. Using these resources, common and rare variants, at least I hope I've shown that common and rare variants are linked to this long telomere type trait that's associated with risk for numerous types of tumors, both benign and malignant. And the only way that what I showed for the latter half could have been done with a data set like the All of Us research program. IME is just one of literally thousands of scientists now who are touching this data. I'm studying a minuscule component of the capacity of the program. There are literally thousands of people who have their own little niche that they're touching this data to study rheumatoid arthritis. And people I'm studying just telomeres in cancer. Some people might be studying rheumatoid arthritis and focusing on everything and anything about rheumatoid arthritis. There could be a dozen people studying breast cancer and causes and factors that you could prevent breast cancer, brain cancer, lupus, who knows, ingrown toenails. All that type of information can be available to understand what causes human disease. This type of information can also be used to hopefully eventually prevent disease. So I don't know if you guys are participants in the program, but if you are, you may recognize that people who have joined in the past had the opportunity to get genetic results back, returned to them. There's some nuance that, and I can't promise any new participants that they can get that type of information moving forward just because of federal budgets. But the program really does want to give back. So when a discovery is made, our participants can learn from those discoveries and hopefully benefit from them. So I don't know if it will ever happen, but I would love to think that someday that one of my discoveries that I made using all of this data might actually go back to the participants who have these rare genetic risks, because then the idea is that then with that type of information, if they choose to receive it, because they could choose not to receive it, but if they choose to receive it, it might empower them to get that personalized precision care that each one of us deserves, because of we're all unique. So, any questions?
Well, we are in the program. I'll just use the tumor one in particular. If through the data search and that, I met all the requirements for the high-risk tumor thing, but I don't have it yet, would my data be kind of, where you're updating periodically, would my data be kind of on a watch list to see if I develop tumors, if I'm in that category?
It's a really good question. So yes, the way the program is structured is that it's designed to be a quote-unquote longitudinal study. So even though you might have enrolled last week, last month, last year, 5 years ago, whenever,
Yeah.
From the time you enrolled, all your historical data that you volunteered to share will be captured, and then twice a year, if you're a patient at Marshall Clinic, we provide that data to the program. So in essence, you are being watched. No one's actually watching you specifically, but yes, your data is going to be tracked over time, and scientists can look at that longitudinal nature so that if you have this genetic predisposition, people can study, well, when would most people with this genetic predisposition actually develop the disease? Is it in the 30s, is it in the 50s, 60s, or 80s? So yes.
If somebody does have those extreme short telomeres, is there any development to how to lengthen those, how to change those?
That's also a really good question. There are pharmaceuticals that have been developed both to lengthen and shorten telomeres.
Okay.
Most of them were designed to shorten telomere length, and most of them were designed for anti-tumor properties. I mentioned when your chromosomes get exceedingly short, your cells are supposed to die. So the idea would be, okay, you have a tumor, if you could target a medication to cause the cells in the tumor to have exceedingly short telomeres, the hope would be the cell would die. Those medications have not worked very well. That's because, at least this is my understanding and what I believe is the likely scenario, is that most tumors have hijacked the telomere machinery to prevent them from ever getting shorter. So even though you're giving them a medication to cause them to be shorter, they already have shortcuts to prevent that from happening. Now, the counter, they've developed medications to make people have longer telomere length. Given this research, I would be actually worried about that. But the underlying biology that is linking telomere length with tumor risk is not quite clear yet. The hypothesis, I'll move over here to this board. Let's see here. Let's go back. Yep. So this is time/age, and this is telomere length. When we're born, I mentioned, you're born with your longest, and then when you die, you die with the shortest, right? There are some people who are born with slightly shorter. They don't always necessarily die, but I mean, there are a lot of factors that can Maybe this person actually lives healthy, and you can imagine their trajectory might look like that. So there's a lot of variability and a lot of factors, but let's say you have a genetic predisposition to be out here. And let's say you have the same lifestyle as this person, and under normal circumstances, this would be what you would see. During your life, your cells are experiencing, each individual cell might have mutations that are being generated and put into that cell. That cancer is a genetic disease. Cancer can be an inherited genetic disease, or it can be a factor of genetic disease that you've experienced because you stood out in the sun too long, or you smoked one too many cigarettes, or you ate one too many donuts, or whatever is the bad thing that can create a mutation in any one of our cells. Under normal circumstances, this normal person, let's say they have a mutation right here that causes that cell to have tumor-like properties, but it hasn't yet developed into a tumor. But it continues to divide, and eventually it's going to peter out, and that cell and all the precursor cells will just die out. But sometimes there might be another mutation that occurs right here that prevents the telomeres from ever getting shorter, and it goes out. Now you have a tumor that's developed in here. Follow so far? This is bad because it had time to develop that second mutation. Now imagine this person, they start off with telomere length that's long, and let's say they have a mutation at the same point in time, and then they develop that second one, they have a more likelihood of their cells turning into a tumor, because they could have a mutation, a second mutation here could develop into tumor, a mutation here could develop into tumor. All these time points in age could develop tumors. But for this person, they run out. These aren't possible because the cells have already died. Do you see? So if you're born with a long tumor length, you just have a greater likelihood of generating these other mutations that might allow the cell to turn into a tumor. That's the acting hypothesis that is linking long telomeres with the disease.
Got a ways to go then.
But for that matter, if I don't know if I'd be excited about taking a drug that causes my telomere lengths to also get longer. So,
Yeah.
but maybe those short telomere drugs might be better in people who have this long telomere condition. So it's failed in almost all clinical trials, but they just were looking at the wrong people. Maybe you design a clinical trial specifically for people who have long telomere length.
Right.
Maybe those are the people who would be more responsive to
Okay.
the medication.
Okay, you haven't been specific yet.
And that's why all of this research program is really important, because it begins to tease out those nuances, those specific factors that cause human disease, which includes at the genetic level, but it can include at the occupation level, at the religion level, at the cultural level, at the environment. Trees. Yeah. Do you have any other questions?
Well, that's the thing that excited us about all of us, generally speaking, is that it wasn't just Marshfield. It encompassed the entire, hoped to encompass the entire country. And where you live has a major effect on our whole growth in that.
Absolutely. So I'll end with this slide. I'm just going to describe for, obviously it's not relevant to you because you're already participants, but for anyone else that listens, how you can still join the program. All you have to do is go to the website and you can set up your own portal. password protected, only you have access to that portal, and you can do all the consenting online in the comforts of your own home. I'll warn you, it can take 2 to 3 hours to do this work, but you don't have to do it all at one time. You can do it laying in bed, sitting in your easy chair, at your desk, wherever. And again, you don't have to do it all at once. Once you've completed that, there is a choice. You can either come into the Marshfield Clinic and get a blood draw, and we can collect your samples and everything like that. That is not going to be lasting forever because of federal budgets. We're going to a pure remote option this summer. But you can do that already if you want to do everything remote. You can get a saliva kit ordered, sent to you, and you just spit in a tube and then mail back, and you're a participant. We hope that our participants willing to share everything about themselves, which includes their health data and some questionnaires. Questionnaires might come out every 6 months to a year. New ones, I don't know if you've seen many of them or chose to do them, but it's really up to you on how engaged you want to be with the program. But it does not require numerous visits or anything like that. So it's really a low-touch project, especially if people share their health record data, because that's really valuable, because all they have to do is go to the doctor, and that information then gets shared with the program. But importantly, all that information is kept de-identified. So even though I have access to the data, I have no idea who is who in the program. And there's a lot of other infrastructure built on to protect participants' privacy. First thing that the program has is a certificate of confidentiality. This is a legal document that is connected to the program that prevents government from gaining access to the data beyond the purpose of research. So if there was a serial killer in our data set, we would never know. Then all the data is kept on a secure system that's password protected, that requires training to gain access to it. So not anyone and everyone can gain access to the data. You have to be a reputable person from a reputable organization that can vouch for you, and then only then, with additional training, you can gain access to the data, and then all the data is kept on the system. I can't download any of it. It's just not possible for me to do that. So I can't make a copy for my own purposes and take it somewhere else. Everything is kept within the system. And that applies to me as it well applies to 1,000 other scientists. What I really love about the program is it democratizes research. Back in the day, before this program ever existed, who were the scientists and who were the organizations that could do research like this?
Pharmaceuticals.
They were pharmaceuticals. They were essentially white, well-educated men like me, but coming from places like University of Wisconsin-Madison, the Harvards, the Johns Hopkins, the Mayos, the Northwesterns, University of Illinois, whatever, Big Ten school or Ivy League school, wherever. Now people such as me from Marshfield Clinic, or even people from University of Wisconsin, Stevens Point, or Eau Claire, scientists who have just as good of ideas, but never had the resources to do these types of experiments, can gain access to the same type of data that everyone else does too. So it really democratizes it, but make sure that all that information is kept confidential so that our participants' security is paramount.
Now you mentioned the federal funding. Are pharmaceuticals also helping to fund the program as a research?
Not yet. There is a desire for that to happen. I don't know when and if that will happen, but we're looking for private partnerships to help maintain the sustainability of the program, recognizing that none of the bylaws of the program and the regulations would ever change because some other entity had access to it. When it was first opened up for science and research, it was only available to academics. They are opening it up to private industries, so long as they have the proper credentials and the proper training. But when it comes to generating new data, we're really looking, that would be where the partnerships could flourish. But put this into perspective, there is a biobank that was before the Aldous program called the United Kingdom Biobank, UK Biobank. And they enrolled 500,000 people into their program. And they did that, I don't know, within the last 10 years. So this is a more mature program. Not to the scope that we have. Obviously, we have a goal of 1 million or more, but we have a lot more types of data that we want to accumulate. But given that they're an older biobank, they've had an opportunity to collect more through these private partnerships. When they did their when they sequenced the genomes of all 500 people, that was done by pharmaceutical companies. But again, all that data was kept confidential. So I could still use the data. I could use the UK Biobank for my own research, but it was initially generated by a private company. I don't know if all of us will get to that, but it's going to be dependent also on that federal funding part. So,
When someone does die, is their history of data still in the bank to be used?
it is, and then when we find out that people die, that's added into it. So
Okay.
people can essentially study causes of death.
Okay, very good.
I don't know if you've heard me talk before, but I presented a study using our biobank of 20,000 where we studied 300 dead people.
Oh.
And we were studying drugs and medications that people took in the context of their genetics over their entire lifetime. And that study, which we published a couple of years ago, showed that nearly everyone carries genes that impact the drugs we take, which is not novel, that's been shown many times before. But since we had the lifelong medical history of these 300 people, we showed that 80% of people were given a drug sometime in their lifetime that was impacted by their genetics. And that type of research could be done then in all of us as well on a much larger scale. So it's something that's on my to-do list.
Very good.
Any other questions? Okay. Well, thank you for your time.